Okay, so I had to redraw my replication bubbles, and they aren't quite equitable, but all right. So now what we have here is I've added the leading strands, and I'm trying to move the screen a little, which I probably should not have done. So in red, I'm now showing the new strand that is being added to the template. So the original strand is in black, the new strand is in red, and the new strand is growing towards the fork because here was the origin of replication. So bases started to get added at that point. We're adding nucleotides along as we go, and these are known as the leading strand. So in each replication bubble, we have leading strands. Okay. Now, because of the way the helicase breaks the bonds, on the opposite side here, you don't get this continuous addition of bases. This at the 3', the new strand. And I will show you, I'll put a link to a video so you can actually see what this looks like in almost real time. But what we end up getting then is what's known as a lagging strand. Okay, so now I've drawn these Okazaki fragments, and hopefully you can see, so 5'. So if this is the 3' end, that's got to match up as 5'. So 5', and we're adding the bases at the 3' end. Okay, 5', 3'. So that's where bases are added. Same on this side. So they're also known as the lagging strand or Okazaki fragments. So the person that discovered these was a researcher, last name Dr. Okazaki. All right, on the opposite, on this bubble, I'm going to add the bases to show you how the complementary strand is being created, or the new strand. Okay, so it got a little crowded in there. So if this were the template, say we had thymine, adenine, thymine, guanine, cytosine, the complementary bases that are being added are adenine, thymine, adenine, cytosine, guanine in here. So a little crowded, but the complementary bases are being added. So I keep saying these bases are added, but how is that happening? Well, there's another enzyme besides helicase that adds the bases. Okay, so polymerase 3 is what adds the nucleotides. So I'm going to use, I've actually run out of colors. I'm going to see if I have a green. Okay, so DNA polymerase 3 is an enzyme which adds the complementary bases to the new growing strand. So I did find a green pen. So it's typically it follows this DNA polymerase 3 follows the helicase. So the bonds are broken, and then once they're broken, the new bases can be added. So we have DNA polymerase 3 here. We have it right behind the helicase here, also here, and showing there. But for the lagging strand, it's again always at the growing, the 3' end of the new strand. So I just outlined that in green there. So that's where the DNA polymerase 3 would have been here, as well as here, because it just adds. Now, once this lagging strand runs into this new strand here, it disengages because it hit a roadblock. I mean, it doesn't have to add any more bases. Okay, so far we've learned of 2 enzymes, helicase that breaks the hydrogen bonds, and then DNA polymerase 3 brings in the new nucleotides. Lastly, I drew the lagging strands on this side of the bubble and showing how the DNA polymerase is again adding at this, again, 3' end. So like this would be 5', because again, here's going to be our new, here's the 3', so 5' would match up, and then 3', 3', 3'. But yes, it's 5' because that's where it started. Okay, the next DNA strand that we'll show will be the complementary bases, basically DNA polymerase completed. Before we can complete the DNA replication, though, we also, you can see, we have these lagging strands. And I mentioned how the DNA polymerase here stops because it's realizes this has already been completed. But we have to glue the 2 ends together and between all the Okazaki fragments. Those are just pieces of DNA. The sugar phosphate backbone is not attached. So we're going to do a 3rd enzyme, and I have pink. This 3rd enzyme is known as ligase. Now note DNA polymerase ends in ase, helicase, ace, and ligase. Ligase acts as a glue, and it glues or attaches, creates the sugar phosphate attachment or bond. It's a covalent bond. It's actually called a phosphodiester bond. So next picture, I'm going to show you the ligase. Okay, I kind of cleaned up my bubble a little bit. So I'm showing the helicase in blue, DNA polymerase in green. Oops, got to move that up. Oh, and I keep forgetting this 5' over here. And now I've added the ligase. It's the black with the stripe. So ligase is glue, which binds. You can't see that, so I'm going to move it up. Oh, actually finished that. Hold on. Okay, so definition for ligase, they always use the term glue. So it acts like the glue which binds the lagging strands together, as well as the lagging to the leading strands. So it creates the bond between the sugar and the phosphate group for the sugar phosphate backbone. All right, so now we have the 3 enzymes, helicase, DNA polymerase 3, and ligase. All right, we do have one final enzyme that I want to mention. It's usually referred to it as DNA polymerase 1, but it's also called DNA polymerase epsilon and delta. And this does a proof checking. It makes sure that mistakes haven't been made, that the wrong complementary base was not added. All right, here is the definition for the last enzyme, the 4th, you should say, is DNA polymerase 1 or polymerase delta. So this enzyme is a proofreading enzyme. It makes certain the correct complementary base is added. All right, so now I'm going to basically show the 2 new strands. Okay, so we started with the parent DNA strand, and it's time for the cell to replicate. So the DNA uncoils, and we have our long, drawn-out, unraveled DNA strand, and oriented 5' to 3', and then 3' to 5'. So each side of the DNA strand acts as a template, or it's the original. And then in the end, what we end up with is 2 new strands. All right, so after creating many replication bubbles, which grow towards each other due to the helicase breaking the hydrogen bonds, the DNA polymerase 3 adding bases to the original strand, creating the new strand, and finally ligase gluing the sugar phosphate backbone together, we get 2 new strands of DNA. So we've now doubled the DNA. And this happens with every chromosome all at the same time. And note, we have an original strand of DNA as well as the new complementary strand. So both new strands of DNA have an original. So because of this, DNA replication, the product is known as being semi-conservative. Half of the DNA, the original DNA molecule, is retained or conserved. So DNA replication produces 2 new strands of DNA, each containing an original side or strand of DNA. This is known as semi-conservative. So half of the DNA remains of the original. Okay, so half have, or half of the original strand of DNA remains or is conserved.